Hello, my name is William Hadges, and I did my project on bacteriophage protein bioinformatics. Phages, viruses that infect bacteria, act as naturally occurring models in genomics and protein studies due to their small genomes and overall simplicity. Phages have genomes that are dense in encoding genes, so they provide ample data for studying the relationship between genetic material and protein synthesis. My part of the project this year involved examining the relationship between structure and function in bacteriophage proteins. Using programs such as PyMol and Fire2, we have been able to accurately predict the structure of a protein based simply on its amino acid sequence. The programs can also be used to analyze comparisons between crystal structures and predicted structures of proteins. These techniques were utilized in the PURC poster project as well. But before getting into that, it's important to understand why we study phage protein structure at all. Having a protein structure provides a greater level of understanding of how a protein works, which can allow us to create hypotheses about how to affect it, control it, or modify it. For example, knowing a protein structure could allow you to design mutations with the intent of changing function. So my work with worksheet 1 involved a PyMol crystal structure comparison of two phage proteins, one OCY, which is a short tail fiber protein, and 3HC7, which is a lysine B protein. The structural differences are quite stark, with the structure of the short tail fiber protein being much more linear and less compact than lysine B. Lysine B contains many more secondary structures in protein folding, consisting of alpha helices and beta sheets that are tightly compacted in a spherical enzyme. The short tail fiber protein, on the other hand, does not display a lot of protein folding or secondary structures. Next, I worked with the Fire2 modeling program to visualize a predicted protein sequence based on the amino acid sequence. This was helpful for working with gene 52 of the draft genome, as the crystal structure is not known yet. This gene codes for a putative DNA helicase, and PyMol allowed for secondary structure analysis that revealed multiple alpha helices that create a series of loops for unwinding DNA. Worksheet 3 involved comparing two different proteins through superimposition of the molecules on PyMol. Gene 58, a DNA primase/helicase from the draft genome, was entered into Fire2 to produce a predicted protein structure, and that prediction was superimposed with its crystal structure. PyMol calculated alignment scores for the match. The two molecules align well, as shown by the RMSD being far below 1, and the match align score being 399.